Hello everyone, my name is Carruthers. I want to thank Sean for the opportunity to come here and talk to you all today. I'm going to try to go through not how to do science, but more about how to think of what's involved in designing a microgravity experiment. A lot different than what you would expect from designing something in normal gravity. You can, if you need to get a hold of me, you can email me or follow me on Twitter, LinkedIn, G+, or Google+, whatever flavor you want. There you go. So who am I? I hate pedigree pages, but they seem to be a necessity. I'm currently working at the Methodist Hospital Research Institute. My day job is I work on how proteins interact with each other in order to turn on and off genes. But I've been involved in aerospace pretty much my whole life. This is a shorter, chunkier me back in probably shortly after one of the single-digit space shuttle missions. I've been pilot for probably 13 years now, and I'm almost always around aerospace in some way, aircraft, either flying them or working on them. I love aerospace. And all my degrees are in biochemistry, but I'm an engineer by heart. I just love taking things apart, building things. If I was to do it all over again, I'd be an aerospace engineer. The other thing is, people sometimes laugh because I put my associate's degree up there, but that's really where I kind of got going. I was pretty lost when I first got out of high school and really didn't know what I wanted to do with my life. And this is really where I kind of got my compass kind of turned into what kind of career field I wanted to do. So it's important for me to put up my associate's degree. So what have I done? I've flown 4 payloads now, starting with STS-91 on 1998. This was a genotoxicology experiment. It's a pretty simple, passive experiment that flew in the payload bay of Discovery. Recently, in 2011, I flew in the last 2 shuttle flights. These were post-crystal growth flights. And recently, and currently actually, I have an experiment that's currently up on the space station as of right now, and should be returning on the 14th. I also have 2 reduced gravity flights under my belt, and that was doing fluid dynamics experiments. So just a brief preview of what I'm going to be talking about today. I'm going to give a really quick overview of areas of microgravity. There's going to be a lot of other talks here today that will go much more in depth into the areas of research. Some hardware for designing microgravity experiments, and then finally just a couple examples of microgravity life science experiments, and I'll go into my protein crystal growth experiment that's currently on station. So what are the most critical areas of life sciences that are going right now? I think there's this picture right here on the Space Life Science website at NASA sums it up really nicely. They have everything from plant growth to molecular biology, studying how cells work in microgravity. They look at things such as microbiology, small bugs, so, E. coli, staphylococcus, etc. And then they studied also the physiology, human physiology, and in other model organisms such as fruit flies, and I have no idea what the snail's up there for. I have yet to figure that one out. It must have been something that flew in space that had to do with snails, I really don't. So plants, why are they looking at plants? Overall, it's for NASA's long-term goal is to look at long-term spaceflight. And we need to have something self-sustaining while we're there. We can't bring all of our food with us, and it's just too much weight. It's just not viable. So can we grow our own plants in space? So their main objective is to look at, can we maintain a certain quality of plant growth in space for food, and do they retain nutritional value in microgravity? This is a really, really awesome facility that they have at the Space Life Sciences building at Kennedy Space Center. And you walk into this thing and you're immediately like in the set of 2001. I mean, you feel like this is what spaceflight is going to be about. This is where we're going to grow plants and how we're going to do it. And they've been doing a lot of awesome work on how to grow plants in 1G, but also looking at how plants grow in microgravity. The other areas are a little bit more specific for plant growth, which has to do with gravity sensing. How does a plant know to grow its roots down or the sunlight go up? Or how does it go up, sense sunlight? Will that work in microgravity? And also as well as cell wall regulation and the genes involved in sensing sunlight and gravity. Molecular biology products are experiments. The microbiology people are going to hate me for lumping them in with molecular biology, but I kind of think that they're very similar. Tends to look at the increase or decrease of gene expression in microgravity, as well as cell structure and cell motility, which are kind of related. Cells have a type of skeleton inside them that allows them to form feet or whatever they need to do to move around. And also as well as to talk to other cells around them. How does that work in microgravity? Is that reduced? Is it increased? Does it change it? Are the cells able to still live without this cytoskeleton, with or without the cytoskeleton? And microbiology looks at pretty much the same thing, but on a single cell or a small colonies of micro living like E. coli or Staphylococcus. And there's actually some evidence that shows that some of these organisms actually become more virulent in microgravity, and that's not good for us, especially if we're looking at long-term spaceflight. Biophysics, which tends to be the area I'm comfortable with. And this looks at, I used molecular biology and biochemistry, but there's an overlap of almost every science involved, from chemistry to physics to genetics. And we usually look at either the structure of protein, DNA, RNA, or viruses, and we use a lot of physical methods to do this. Some of the other things that we tend to look at are enzyme kinetics. How do enzymes create a product? How fast do they? How slow do they create this product? Does that change in microgravity? We also look at protein interactions. Exactly how do these interactions occur, and does that change in microgravity? Physiology is looking at, essentially, for NASA tends to look at human organisms, but like I mentioned before, you can also look at model organisms such as fruit flies or worms. And you can look, there's a significant loss to astronauts in long-term spaceflight. They lose about 1.5% to 2% of their bone mass per month in space, which is huge. Almost, I think it's 50 or 60% of astronauts upon getting into microgravity have this space sickness, and nobody knows why. Usually might have something to do with vestibular confusion in their ear. Cardiovascular changes or orthostatic changes. All your fluid in your body tends to go from your legs all up into your upper core and in your head. If you looked at that video of Chris earlier, I don't know if you mentioned it and watch it again, you'll see his watch is around his wrist. And it's really, really loose. But it was tight when he got up in space. So it's like, since he's been up there, he's lost so much fluid that it's actually really loose around his wrist now. Vision changes is another big thing. Due to the pressure changes that I just mentioned, your eyes can actually change the way they focus. And this is a big problem when you're in space and you can't see what you're doing. They actually have developed types of glasses that astronauts can wear to actually change the focus of the lens so that they can see better. And there's actually a very small percentage of astronauts who return back to space and their eyesight was screwed up significantly. Immune system changes are pretty significant as well, probably because all of the above is happening in your body and your immune system is trying to fight off all these quick changes that occur to you. You can study pretty much all of these in other model organisms, as I mentioned earlier. They're probably a heck of a lot cheaper than humans and less paperwork. We'd already started discussing in the last talk about suborbital vehicles. All my experience is on orbital, which is space station. But everything that I'm going to talk about can be directly applied to suborbital. And suborbital is critical to learn how your experiment is going to work when you do go orbital. They have a quicker turnaround time which was, I think one of the questions earlier was discussed about how quick can I get my research on. As scientists, we want to replicate what we've done. We want to keep going with our experiments. And science moves really, really fast. And we get a result, we want to change the next experiment and keep going. So we're always pretty concerned about how quick can we get on to the next experiment. The suborbital is also going to be a shorter microgravity time, but like I said, it's going to be compiled over many different flights. You can get a lot of data really quickly, unlike suborbital, which while there's a much longer microgravity time, there's usually less flights. There's usually at least 3 or 4 flights a year, maybe. There's also the cost value. Suborbital is going to be significantly less than orbital, which is, I think, a factor for many of us. But still, it's a great tool to learn how your experiment may work if you do plan to eventually go to orbital research. So how about some hardware? What I'm about to go over, most of you probably are somewhat aware of in this room, I'm pretty sure. So I'm just going to go over really quickly about some of the devices that are out there. We live in a really awesome time to do experiments now because there is so much available to just general citizens out there to build your own equipment. It just blows my mind when I go to some of these websites like Adafruit Industries. It just blows my mind how cool this stuff is and what's available to us as far as researchers. So this is my mantra for whenever I'm thinking about my next experiment. I'm always thinking about what I'm going to be doing next. Compact, safe, and automated. How small can I make this? Can I make it really, really small? Can I put it in a cube? Can I put it in a 1U, 2U, 3U, etc.? What's the smallest amount that I can put my experiment in? Safe. If you're going to be flying this with people in the aircraft, that's primarily what you're going to want. Now, you're not going to want this thing to blow up. You're not going to want it to open up and expose the astronauts to anything that you're growing. Second of all, vehicle safety. You don't want this thing blowing up. You don't want to be on the news as the one that blew up the space station. It would not be a good thing. It would not be a good thing. And automated. As I mentioned earlier, it's so easy now to automate your experiments. And while there's a romantic view of I want astronauts to do my science and all this, there's a lot of variability between astronauts. And some of them are engineers, some of them are pilots, some of them are scientists, some of them are busy that day and they really just want to get through your experiment. Some of them, they're just like people like me when I'm in the lab. Sometimes I just want to get through my experiment. Sometimes I want to actually learn more or do more. So there's going to be a lot more variability in your research if you have an astronaut involved in your data loop. So try to make it as automated as possible as you can. When doing a life science experiment, it's the same as what's called an ECLSS, or Environmental Control Life Support System, that NASA uses. It's the same thing. It's the same thing requirements that you want, except for your little cube experiment. You're always going to want to look at, do I need temperature requirements? Are there fluid requirements? Do I need light requirements if I'm looking at plant growth? And air requirements. Do you need to remove CO2? Do you need to put in oxygen? These are things to always think about when you're designing some sort of life experiment. How can I keep my life science experiment alive for the allotted time that it's going to be running? There's no Portal fans in here? Nobody got that? Some Fanny Cuba. Something I don't see a lot of is the use of these Microtiter plates. These things are awesome. They're made of plastic, or variable different types of plastic, variable colors from black to white to clear. They come in a variety of sizes, anything in multiples of 6. So you can put whatever you want in each one of these individual little wells. They come in, some of them are 96 with 2 mil volumes. They come with a sealing plate, like a sealant that you can put on top. Some of them have clear bottoms, so if you have an experiment that you want to look at, say light creation of you have a gene that creates light when it's turned on and you want to measure that, you can measure that directly through the plate. And they all have a standard footprint. Even though they have all these multiples of 6, they all have the same size. And this will easily fit in a 1.5 U cube lab. It's pretty rare that you're going to have a life science experiment that you don't have to do with fluids. And some of the best things out there for these things, and they're really cheap, are syringes. You can hook up a syringe to a step motor and push in or push out how much fluid you want in your research. Peristaltic pumps. There's this little itty bitty peristaltic pumps, which are essentially just a little DC motor with 2 little knobs on them that move fluid through a tube. Pressurized fluid containers. Essentially, I don't know, once again in that video you saw earlier where Chris was squeezing the bottle and the water comes right out. You can do that. You can pressurize your container beforehand, and as soon as you open a valve, the water just comes right out. They're a little bit more paperwork because they're a pressure vessel and they have to be tested, not that they're going to explode, but it's worth it. Agar-enriched media is a good one. You can easily take agar from a lab, mix it in with some sort of growth media, whatever your E. coli or your plant is going to use, embed the seeds or the E. coli into that. It'll use the media from around it, and it also keeps the fluid all in one space from moving around. It's like Jello, actually. Capillaries are great. Water always wants to, depending on its surrounding material, always wants to flow with water. So if you can have somewhat of a passive fluid moving system just by having capillary, by changing the diameter of the tubing. Surgical and aquarium tubing is available everywhere. This is great for moving your fluids from syringes or pumps to your experiment. I don't really have to say that much about LEDs. They've been around forever. Even my grandmother knew what LEDs were. Variety of sizes and shapes. What you may not know is the variety of wavelengths that they're available. And you can get these from infrared all the way to UV for anything that you want to test. That's incredible. And they're relatively cheap. So how about temperature control? Probably the most you can if your experiment can deal with it, just ambient is okay, but definitely find out what the ambient temperature or fluctuations are going to be on the vehicle you're flying on. ISS tends to be about 22 to 25, but it varies quite a bit. You can use a heat sink to move heat away from whatever, if you have a microboard inside your experiment to remove some of the heat. You can use some of the active things you can use is such a fan to remove move air through, either bring in cooler air, warmer air out. Peltiers are awesome. If you know what a Peltier chip is, essentially a current goes on one side, it's two types of metals. It can heat up one side and it keeps the other side cool, and these things will actually build ice on them. They get so cool. And they're relatively small. This is only one size. They go up pretty big, and they actually get smaller than this. Air, once again, if you could use just passive atmosphere, find out what kind of atmosphere is going to be involved in the pressure in the capsule or ISS or whatever you're going to be in. If that's okay with your experiment, that's good. It's passive. Or if you need some sort of air pumped in, do they have air such as carbon dioxide, oxygen, nitrogen that you can either add or that you can add to your experiment? And if so, find out what the flow rate of this is and if it's conducive for your experiment. Detectors. These are some of the detectors, some of the things I was talking about that Adafruit that kind of get me excited. These are so small. I mean, they're itty bitty sensors. The camera, this is a 3-axis gyro, barometric pressure sensors. Everything you can think of to put in your experiment to measure something. You can get data going directly to an Arduino, straight to an SD, and you're golden. It's awesome to me. And also another good source of a lot of this stuff is old smartphones. Look around and see who's got some old smartphones laying around. Grab them, take them apart. There's little cameras in them, there's accelerometers, there's little vibrator motors in them. They're great for building into your experiment. So like I said, when you're going to design your experiment hardware-wise, the mantra that like I said, always compact, safe, and automated. And then learn about as much as you can about the vehicle that you're going to be flying on. Learn about as much as what kind of conditions it's going to be going under, what kind of G-forces, what kind of temperature changes, et cetera, in order to build your experiment to get the best data possible. So what are some examples of microgravity life sciences? I'm going to go through two really quickly, not about the science, but more about the design that I thought were really awesome. And then I'll go into the protein crystal growth experiment that I'm working on. This is one from Nanoracks from Valley Christian High, your neighbors here, not too far down the road. This is a plant growth experiment, and it has a little enclosed capsule here with 6 little pods that have they planted seeds in. There's a pressurized water container back here that has a valve on it. It has 12 LEDs for lighting of the plants when they grow. And it's all controlled here by a microprocessor board. The microprocessor directs the light to go on and off, so, day and night, to simulate day and night. And it also opens and closes the valve that's contained in the pressure vessel in order to water the plants, essentially. This is I believe it looks like it's about 2U form factor. It could be 2.5, I'm not sure. Is it 2U? This is another one that I think is really awesome. It's not a cube form factor, but I think they've packed so much in there. It follows the mantra I was talking about of compact and safe and automated. This is an experiment called Eostio, which flew on a Russian vessel for about 12 days, just went up into orbit and then came back down. So this experiment is completely automated, run off this microcontroller board over here. And these little 3 cylinders, there's slides that have cells on them, bone cells. And they are in a media to make them happy, and they have these syringes all lined up here, which they can pump in media, fresh media, and out comes the old media into this bag up here. There's another couple syringes for fixing the cells. There's one that has actually hormones in it to give to the cells to see how they react to the cells and the hormone and microgravity. There's a rinsing solution here. But this is all about the size of a medium pizza box. It's not that big at all. They get a lot of science out of this little itty bitty box. And it all runs automatically. I believe it had a sensor in it that once it sensed an accelerometer that once it sensed a certain amount of time that went by without in microgravity, that it turned itself on, which I thought was pretty sharp. So now getting to my comfort zone. Why do we study biological macromolecules at all? And this is a saying that was from one of my mentors at Texas A&M, and he just said, he said this often, it's kind of funny. Life is just about protein interactions. Every living thing doesn't exist without some sort of protein interaction. DNA is important and all, but without proteins, DNA doesn't survive. Somebody from the DNA side would probably argue that with me, but so the way we study the structure of proteins is using a technique called there's several methods of studying the structure of proteins. The method that I use is called X-ray crystallography. And it's a powerful method of looking at an atomic resolution of the structure of the individual atoms within a protein, or DNA, or an RNA, or a virus. And it gives you a much deeper understanding of the mechanisms of life itself, to which we can look at how, at a molecular level, how a protein can turn a molecule from one thing into the next, and that molecule can be incredibly important for keeping you alive. So the first thing we do in order to study the structure of protein is grow protein crystals. And these aren't crystals like salt crystals, these are more like jello crystals. They're really soft, they're 40 to 80% water. And we have no idea what will make a protein crystallize. We really do not have a clue. That would be the holy grail of all structure, X-ray crystallography. We could look at a protein amino acid sequence and go, based on this amino acid sequence, we know it's going to grow a crystal. We have no idea. Why do we grow a crystal? Because the proteins are incredibly small. That's actually one of the reasons we use X-rays, is because the wavelength of the X-ray is about the size of a carbon-to-carbon bond within a protein. So this is right at the perfect wavelength for us for looking at these protein structures. What it does is if we literally just took one protein and put it in a crystal or an X-ray beam, it just blow it apart. It just wouldn't last very long. Secondly, if we get any data from that at all, most of the X-rays, probably close to 100% of it, would just go straight through the protein itself. So what we do is by crystallizing is we form a really concentrated area of proteins into one small little area. And what happens is the X-rays, when the X-rays go through it, is they create this little pattern here called a diffraction pattern. And what that is those are X-rays bouncing off the atoms within a protein. And this is the only data we get. If you look really closely up at these dots, this diffraction pattern, it creates itty bitty dots. This is the only data we get, and it's important, incredibly important, that the quality of this data is the best that it can be. So the crystals have to be the best they can be in order for us to get the best data we can. Garbage in, garbage out. If you put in garbage crystal, you're going to get garbage data. So based upon this diffraction pattern, we use a lot of fancy math and computers to give us what's called an electron density map. And within the electron density map, we know what amino acid structures are. We can actually take an amino acid chain and form it into this, using computers once again, fit it into this electron density. So everything stems upon quality of the crystals, that we grow, and the diffraction data that we get. But the problem with growing crystals in 1 gravity, in 1G, is that they can have a lot of irregularities. There can be convection and sedimentation within a little drop that we grow the crystals in, meaning that convection is like a hot air balloon, and sedimentation is much like oil and water separating. Due to gravity, something denser will sink and rise. And what this does is it moves the crystal as it begins to grow through different areas of concentration within the drop. And as you grow, you move through these different concentrations, the rate at which the crystallization process happens can increase and decrease. This creates irregularities. This is a technique called atomic force microscope. And what you can see in each one of these is actually a protein. Not much resolution there, but you can actually see protein after protein after protein lined up to form a crystal. But what's missing is there's these little gaps. And what happens when you shoot an X-ray through those gaps is it can actually change the spot into spot overlapping or spot smearing. So it can ruin our data, essentially. There's also this other areas where you can have, this is not as the same resolution as this picture here, but you can actually get areas that grow in different areas of I guess you could say the speed of growth within the crystal. And right here, you actually have a crystal, little microcrystal around another crystal. So you can have all these deformities within one gravity, or 1G, I should say 1 gravity, 1G when growing crystals. And one of the things that you don't have in microgravity is you don't have convection or sedimentation. And you don't have the crystal as it grows moving through all these different areas of concentration. And so therefore, the protein grows at a much more uniform rate. And that crystal, that growing at a much more uniform rate creates better data for us. So how many, just a guess from anybody, how many proteins have ever been flown in space in microgravity? Anybody know? Anybody have a guess, stab? Is it a lot, a little? A few, 2, 3, 100?
A few.
Huh?
It's a sport pharmaceutical reasons.
How many have flown? Over 25 years or 30 years of shuttle, how many have flown? 270. That's it. There's millions of proteins in our body, millions. We have not even begun to look at how many, to look at the structure of proteins from crystals grown in microgravity. A lot of the experiments flown were for improving the crystal structure. They grew okay on Earth, but we wanted to see if we can improve the data quality. And there was a lot of research just in looking at how do crystals grow in microgravity. And within that 25 years, the technology to grow crystals had reached a really significant level of maturity. It was pretty good. But it stopped around 2000. And what happened around late 1990s, 2000? Space station happened. So pretty much all the resources for NASA went toward building the space station. A lot of funding for life sciences actually diminished. So crystallography in space kind of remained stagnant. These are some of the crystals that were actually grown in microgravity, and these are some of the models that they made based upon that those better crystals that they grew. That's another, most people don't know exactly if they actually ever got anything from the crystals that grew in space. They actually have. This is only a small sample size of those. One of them was respiratory syncytial virus, which causes pneumonia. They actually were able to grow better antibodies for this disease in order to study it better. Sharing plow is a really interesting story about their interferon protein, in which they were able to make better much more effective hepatitis C drugs from microgravity crystals. And very, very recently, the Japanese Space Administration was actually able to grow crystals in microgravity where they saw extra atoms that they had never seen before, and these extra atoms also allowed them to make a much more effective drug that had never been available before. Much more specific drug, actually. So this is from a database that's online called the Protein Data Bank. And it's pretty much from anything that's funded by the National Institutes of Health. If you have a crystal structure, you have to deposit these coordinates within this database. And you can see how it's increased over time quite a bit. Actually, a lot, actually. And right around here in 2000, there was between 2000 and 2010, there was an incredible leap in technology in protein crystal growth. And what that was is there was a high throughput, what they call high throughput methods, when you could actually screen different growing conditions for crystals in the thousands. It's routine for me now in my lab to set up 1,000 conditions in 15 minutes to see what conditions will make a crystal grow. Routine. Back in the 1990s, you'd do maybe a couple hundred conditions. So due to this low volume, high density, high throughput as they call it, they were actually able to incredibly accelerate the amount of structures that have been solved through But since the funding cut and pretty much the cessation of protein crystallography and microgravity research, the equipment remained stagnant. The equipment didn't change either. So when it became time for Iss utilization, all they had was the shuttle hardware. Nobody had any new hardware. Everybody was using this old hardware that I actually had an opportunity to use on the last 2 shuttle flights. And I was just amazed. I'm like, this is really a lot of protein, my precious protein that you spend hours purifying and making and is expensive to get. They wanted these huge amounts of it. I felt like I was back in the 90s doing crystallography. So they wanted a large sample size, and you only got about 3 different conditions you could grow it in, 3, 4, 5, however much your wallet could afford, really. So I wanted to know, is there a commercial off-the alternative for microgravity? Is there something out there that is very common to a researcher in a lab that I don't have to be trained upon, I don't have to have any additional training on, that I can just pick up, NASA could have, or anybody could use that I was already familiar with in a lab? A lot of the hardware they had was designed by engineers. It took a little bit of training to learn how to use it. Is there something out there that we can just take right off the shelf and use? So I had seen something called an Pcs Plug Maker a couple of years ago, 3 years ago. And I'm always thinking about, whenever I see devices, will this work in microgravity? How will it work in microgravity? I'm always thinking. And I immediately thought that this would be an excellent alternative for growing crystals in space, because it reduces the need to deal with fluids. It's in a little microcapillary tube. I'll show you in a second about it. It increases the throughput of the research, the experiments, and it also decreases the amount of protein required. I'm just going to show you a little video really quickly, and then I'll describe what you're seeing. Here's the blue. Oops. There it is. There it is. So the way this works is it's a microfluidic design. You can. I just ruined that. I sure did. Get back to that. There it is. You have 3 reservoirs for buffer, a precipitant, or your protein, and it flows. It's connected to this little card here. This card is about the size of a microscope slide. And it creates these little plugs that are 20 nanoliters, 20 billionth of a liter small, in this card. The entire diameter of the capillary is about 200 microns, and you can set up between 400 to 800 of these plugs on a size of a microscope slide. And you only need about 4 microliters of protein, 4 millionths of the protein. So most of the stuff on shuttle was, I think the smallest they had was 5 microliters. But usually it was between 50 to 100 microliters. 100, incredibly, it's several orders of magnitude smaller, this technique. And the way this machine works is you load up your protein and your buffer and what you need to do the experiment. There it goes. So this is the same setup we saw earlier. Pretty much describes itself. And it's now filling these little plugs with a ratio of precipitant to buffer, which you can control, or protein. You can control either one. You can change the ratio of them to create different growing conditions for your crystal. And within 30 seconds, maybe, 30, 45 seconds, you fill up the whole card with 400, 800 conditions. Individual experiments on this card. And what it's doing is you can actually see there's a smaller amount of blue compared to the clear. So it's actually changing the ratio of the precipitant to the buffer. close up. Yeah, close up of the plugs. It's usually not blue. They colored it for better visualization. This is just zooming into the plugs. I'm almost done. And then it shows a time lapse of actual crystal growth. They don't grow that fast, but I wish. This is the machine. This is the slides. Close up of the slides. This is a little crystal. You can take 4 of these slides and put it on a rack. This is just the same exact size as a 96 microwell plate. And then you can take 25 of these I guess they're called forms or whatever, and put them into these boxes. These are both 1.5 units, 1.5 unit boxes. Not even the size of a shoebox. Wow. So this is our workflow. We fill the cards with whatever proteins you need, and we immediately freeze them. And this is another benefit. You dip the card in liquid nitrogen, and it's frozen until you need to send it up to the space shuttle. So you could literally, or space shuttle, space stations. You can literally freeze this for as long as you need to, whenever your flight's going to fly. Launch the space station. You give it to Smiling Rex Walheim here at the space station, and then they thaw out. They're frozen. They're kept frozen in a freezer. Then you can survey them with a little USB microscope that's available through NanoRacks up on the space station. And then you can get them if we haven't tested this yet. It's what we're doing next. But there'll be a possibility to return your crystals. But you can actually see that if your crystals have grown up before you even get them back, which is another benefit, because what if they don't survive re-entry? Keep forgetting you have a button. So as of last Monday morning, Chris Hadfield got to be my slave for a couple hours and do my experiment up on space station. And our preliminary results look incredibly promising. So our primary goal of this entire experiment was just to see if the crystals will grow using this method. And we pretty much beat them up. We put them in this little microcapillary thing, we froze them, we sent them into space. Will this even work? The proteins are nothing too exciting, but they're pretty much standards that we use, but it's a test. It's a test flight. The samples come back on May 14th, and hopefully they'll return. I'm happy about the results we've already gotten. So this is just some resources. If anybody's got a camera, they can zoom in and take a picture really quickly, or ask me later for this slide. But this is a lot of stuff that has inspired me for designing experiments. And I just want to give a thank you to Rolando, Sean for letting me come and talk, NanoRacks, and my boss, Paul Webb.